This is a production of Cornell University.
Nice to see everyone, and today I'm going to be talking about my master's research on the apple microbiome. All right, and so I want to start by posing a large-scale question of how do we have fresh fruit and vegetables, fresh produce in the grocery store for so long after harvest season for many of these crops? It's one of those things where I just kind of took it for granted growing up, and it wasn't until I started here that I really began kind of asking questions about the science of how that happens. And one of the important things to recognize is that a lot of the fruits and vegetables that we harvest don't actually make it to the grocery stores, don't actually make it to consumers. And 20 to 40% are lost after harvest before making it into the hands of consumers. And this is especially relevant for developing countries that don't have the same technology and of wide-scale refrigeration that we do, right? And so this is a very important issue. And the purpose of post-harvest biology is to go in and solve this issue. And the way we think about this scientifically is asking, all right, so what are the different aspects, different forces that make produce go bad? And then how do we stop that from happening to keep produce staying good? This is not an exhaustive list of everything, mostly trying to hit the big points of what we do in post-harvest biology. So I'm going to go through some of these a bit more, starting with ripening and senescence, that the more plant tissues respire, the faster they ripen and senesce, because that's what controls a lot of metabolism. Right. And so there are a few ways we know to slow respiration. Refrigeration is a common technique that helps out with many of the issues that we have. And a couple technologies that I'm going to be talking about in particular that I'm going to be using in my research are low atmosphere controlled oxygen storage and reducing the production and perception of a hormone, ethylene. All right, so first going into controlled atmosphere storage. So the point of this is that instead of just storing produce such as apples in cold air with 21% oxygen, we can artificially lower that down to 0.5 to 2%, depending on the cultivar, and elevating CO2. And the main reason we do this is because oxygen, as we all know, is very important for cellular respiration, acts as the final electron acceptor. So if we reduce the concentration of oxygen, we reduce the rate at which cellular respiration happens and slow down metabolism in general. And one of the aspects that I'm not going to be directly working with here, but I think it's good to mention, is a new improvement on this technology, dynamic controlled atmosphere, that allows us to potentially take oxygen levels even lower and maintain high quality, because the reason we don't bring oxygen all the way down to zero is, if you remember back to basic biology, that there's no oxygen, cells will undergo fermentation and burn through their sugars very quickly. And that's not what we want. So dynamic controlled atmosphere allows us to monitor the apples to ensure that that's not happening while keeping the oxygen as low as possible, essentially to minimize the overall level of respiration. All right, and then another important aspect of ripening and post-harvest, especially for fruits such as apples, is the hormone ethylene. So ethylene is a hormone that has a variety of different effects, and the most relevant one to post-harvest physiology is that many fruits called climacteric fruits, such as apples, will at a certain point in their development produce a lot of ethylene very quickly. And what that does is that stimulates respiration to go up for this time. And so if you can imagine, if we cut off this ethylene production, we can smooth out this respiration graph and lower the amount of respiration, maintain the fruit's quality for much longer. Especially important for fruits like apples compared to something like banana, where we want some ripening after we pick it. We don't want to eat just green bananas, but with apples, we prefer them to be exactly how they are after we pick them off the tree. All right, and so then, of course, the question is, great, we would love to inhibit ethylene. How do we actually do that? And there are a couple different ways that I'm going to go through talking about how we do that. So here I have the ethylene biosynthesis pathway, where we have this chemical SAM that's converted to ACC and then oxidized into ethylene. And the two chemicals that I'll be talking about, one that I'm using in my experiment, AVG, basically stops this synthesis pathway. So you cut off the synthesis of ethylene, make it so that ethylene concentrations are much lower. and we don't get that climacteric, this increased respiration. And, or at least the climacteric is greatly reduced. And the other way, one of the other major ways you can reduce ethylene chemically is with a compound that the Watkins lab has been investigating for a long time, 1-MCP. And that's a compound that I am going to be using in some of my PhD work, looking at the effects of that on the microbiome. But for here, for my master's work, I'm mostly just focusing on AVG. But 1-MCP is still very important, and it doesn't directly reduce the synthesis of ethylene, but it does stop ethylene from affecting the cells in ways that would increase respiration by stopping the perception of ethylene. And then the next point I like to walk people through is reducing water loss. No one wants to like go in and eat like a super shriveled apple that's lost all of its water, right? And so how do we prevent that from happening? Again, refrigeration, and then if you pack a bunch of fruit together, there's less surface area. And then one of the main ways we reduce water loss is by trying to keep these commodities in high humidity, right? And I have a picture here of apples with a bunch of water on them, and that for anyone who's worked with fruit or vegetables, this should probably set off warning signs. If you have water all over your fruit, that's kind of dangerous. And why is that? Not that apples on 95% humidity actually look like this, but the principle applies that if you have a lot of humidity, a lot of water, it can be kind of dangerous for the fruit because of post-harvest pathogens, right? And so a lot of us are familiar with pathogens that affect apples and other fruits and vegetables in the field, and many of these pathogens can also affect apples during storage, right? Something like bitter rot you see here affecting an apple in the field, pretty big problem during storage as well. And we can control these molds and rots a variety of ways. Refrigeration helps with many of them. We can also use fungicides, but those do have certain limitations. There are certain regulatory concerns around conventional fungicides, not that there aren't around other types of fungicides as well. And also it's becoming more and more difficult to discover new modes of action for fungicides. So certain researchers have thought about, okay, what are some other techniques we can use to help control these post-harvest rots and molds? And one of the ideas they've settled on was biocontrol, where basically the idea is instead of just spraying a chemically synthesized compound to some compound synthesized in a lab to go off these post-harvest pathogens, you add on live organisms to antagonize those pathogens or products from those organisms. And it sounds kind of great in theory if it works. It's like you don't have to spray all these like chemicals that consumers don't like on the fruit. You just put the microbes that chase off the bad microbes. But there are some limitations to this approach as well, that it only has full efficacy under certain conditions because you are working with, in many cases, live organisms. So their ability to actually control the pathogens is much more context dependent. And they're not always quite as commercially competitive compared to conventional fungicides on a large scale, but there are still commercial options available and people are working on trying to improve them. And then one piece that many of these post-harvest biocontrol researchers are thinking as could potentially be the missing link is the microbiome, right? Because we think of the traditional way we think of biocontrol is you add a microbe to the fruit surface and it will fight off the pathogen. But the truth is anything we see on the fruit surface, there are lots of different microbes there. And the way I like to think about it is that the fruit surface is kind of like a tiny forest. And that if a forest, if there's one species under control, if you add in another species to try and control it, taking lessons from basic ecology, you need to make sure that the biocontrol species you're adding in not only can fight off the organism you don't like, the pathogen that's causing problems, but also that it fits well into the context of a local environment, right? And so that's somewhat difficult to do because we don't know what a lot of these microbes do functionally. But it's an exciting new frontier for people to explore and could potentially, in the long term, lead to greater efficacy for biocontrol as a real alternative to conventional fungicides, right? And then another piece of theory for why people put so much stress on this microbiome, fruit surface microbiome, is taking from the human microbiome studies that developed this theory called the holobiont theory, which basically states that the way humans work as a individual is not just based on human physiology, but the interaction of human physiology and microbial physiology. There's been a lot of amazing work from the human microbiome world with pretty stunning results, such as the fact that gut microbes can release neurotransmitters that can affect brain signaling. So even the fundamental ways we act as a human are sort of in conversation with the microbes that live in and on us. And so people in the post-harvest physiology world have thought, okay, why don't we apply that to fruit surfaces? That it would make sense that because microbial ecology is so complicated, that everything we think about of how these fruits actually function in storage is a conversation between fruit physiology and microbial ecology. And that's going to be one of the big ideas behind my research. Right. And then as a bit of a fun side note, that Jenny today brought in this apple for demonstration, and a fun thought experiment is, well, when we actually eat an apple like this, how many bacteria are we consuming? All right. And then looking at, there was a study that investigated how many bacteria there are on per gram of different parts of the apple, looking at the stem, peel, the flesh within, and then the seeds at the very core. So I'm not going to call on people like it's a class, but I want to give everyone a second to think, how many bacteria do we expect there to be per gram of fruit tissue in these different parts? So I'm just going to wait a few seconds to give everyone a little bit of time to think about, what's what ballpark? And
10 to the 8th.
10 to the 8th, and some of those are, yeah, well, in the stem part and the seeds part, that's very close per gram. So congrats to Bill Miller on that. And then we see there's
26 and to the 8th.
And we see that there's a lot of variation in different parts of the apple, and that some of this I feel like was surprising to me, but somewhat intuitive, that the woodier parts of the tissue, like the stem, have more bacteria. You wouldn't want your kid eating that or putting their mouth all over it. It makes sense that we have that impression because there are lots and lots of bacteria on it. All right, and so like I talked about, one of the key ideas of my work is combining the microbial ecology I've been talking about for the past few slides with the post-harvest physiology problems that I was talking about at the very beginning. Right. And so that leads into my specific research question: how do AVG and control atmosphere storage affect the apple surface microbiome? Right. And I'm going to go through again what these two technologies are and what we expect them to do first. So AVG, which can come in the form of this powder that you dissolve in a liquid and then spray on the fruits pre-harvest, is an inhibitor of ethylene biosynthesis. So it stops ethylene from being synthesized, which slows down ripening and senescence. And the question is, why would we expect that to affect the surface microbiome? Well, as I mentioned, ethylene is a plant hormone that has a variety of functions. And we know that a couple important things we know about ethylene is that it does modulate plant pathogen interactions. So it's not a huge stretch to say, well, there might be something there in how it interacts with the whole microbiome. And also we know that certain microbes, such as certain bacteria, can synthesize ethylene and use it as a potential signaling molecule. So interrupting ethylene dynamics could have some pretty drastic effects on the microbiome. And then with controlled atmosphere storage, it seems kind of intuitive to wonder, okay, so the point of controlled atmosphere storage is to slow down aerobic respiration. Wouldn't that affect the microbes on the surface as well as the apple itself? And we know that certain studies have shown that it does suppress pathogen growth, not that it directly kills off pathogens for the most part, but it does stop them from growing as quickly, and it can interfere with biocontrol, that if you put a biocontrol organism on, that it doesn't always behave at full efficacy when it's not able to use respiration at full efficiency and grow to actually antagonize the pathogen. And so we're wondering, how does this impact the whole microbiome? And then since this is master's defense, we want it to be kind of very meticulous about making sure that our whole hypothesis is clear to everyone. So our hypothesis is that AVG will shift the microbiome due to, as I mentioned, ethylene modulating plant microbe interactions, and then also ethylene being a bacterial and microbial signaling molecule. And then we predict that a control atmosphere will create a different microbiome than air storage due to the low oxygen, and then this will be more pronounced at lower concentrations of oxygen. All right, so now I'm going to go a bit into my experimental design, where one of the difficulties of looking at controlled atmosphere compared to air is the fact that we weren't able to have the same sampling times for every treatment because we were interested in looking at controlled atmosphere at the very end of its lifespan. But we can't air store apples for that long using our methods. So we have 3 and 5 months air storage, and then 5 and 9 months for our 2 controlled atmosphere treatments, 2% and 0.5% oxygen. And then to give a sort of like broad overview of what we did to actually get the data we're working with, and I'll go into a bit more detail with some of the steps. Then we take our apples, 10 apples per sample. We are able to put them in a wash solution and get the microbial pellet from them. We are able to extract the DNA and then amplify a known region to get these amplicons that are then sequenced, and we use bioinformatics to get a big multivariate feature table. All right, so let's go through some of that in a bit more detail. All right, so a lot of times when I get questions about like how do you actually get the microbes off an apple, there are different ways you can do it. The method we used that we found was most consistent, and was you basically put the apples in a bag with a wash solution containing a surfactant, basic salt, use a sonicator bath to use the sonic waves to knock off the microbes, and then use this rotary shaker to just shake the apples around in a bag, which looks kind of funny when you see it for the first time. Take the solution, filter it through using vacuum filtration, and scrape off the grime, and that is our microbial pellet that we use a DNA extraction kit on to get the DNA that gets sequenced. All right, and I talked a bit about this in the overview slide, but the method of sequencing we're using here is Amplicon sequencing. So we're not going through and sequencing entire genomes of entire bacteria and fungi because that's very expensive, especially to do for an entire microbiome. What we're doing is we're taking part of a known gene. If you ever heard of 16S or ITS sequencing, that's what this refers to. Do PCR to amplify, to get amplicons, many copies of that region for each DNA strand, and then sequence those to then see what those microbes are. If we can map them back onto reference genomes later. And the upside of this is that we can use this and get a lot of samples, so we can actually do stats. But the downside is we don't always get species level identification. So that's one good thing to keep in mind with all the data I'm going to be showing, is that we get a general idea of who's there, but not down to the species level, which can be impactful. All right. And then again, because this is the master's defense, part of the master's defense, we decided it was a good idea to be pretty explicit about a lot of the different methods we were using, even if it gets a little bit technical at times. So I'm just going to ask you to bear with me. So once we get our actual DNA sequences back, what we do is we use a program to do a bunch of filtering. We merge the forward and reverse reads. If you have done Illumina sequencing before, there's a good chance you've worked with that, merging reads. And then you get these, what are known as ASVs, so, which gives us a table of, okay, here are all the different DNA sequences, and here are how many copies we have of each one. What we do then, which is optional, some people like to do this, some people don't, is say, okay, all of these DNA sequences with 97% similarity, we're going to basically regard as the same thing, basically the same type of microbe. Not quite a species, because again, we don't always get species level identification, but something kind of similar that is not species specific necessarily. And so we then get those what are called operational taxonomic units, or OTUs. And we can use that table to directly perform stats on. We can classify the taxonomy for them. And then we can also, I'll go into a bit more of this later, predict the gene pathways of those reference genomes, which can potentially tell us more about what those bacteria are doing. For bacteria only, we unfortunately don't have the program doesn't work for fungi, at least yet. All right. And then going into some of the stats parameters as well, in case anyone is interested in exactly how I did this or wants to work with microbiomes in the future. One of the big debates in the field is whether or not you rarefy, basically cut out data until every sample has the same number of reads, the same number of microbe counts. And I chose not to do this because the argument for doing this is, well, you want to make sure like each sample has the same amount of data so you can actually compare them. But my stance is that it's better to just use kind of the full data set to make each sample the most accurate it can possibly be. And then we basically, as a result of this, because we're working with a large ecological data set, we get this huge multivariate data frame. And the way we calculate statistical tests on what's different is using this Bray-Curtis dissimilarity matrix instead of a standard like Euclidean distance where you would just subtract a bunch of things. Basically, the functional difference here is that it handles zeros better in the data set. And then we were able to do a lot of multiple testing to find which gene pathways and which genera are significantly different between some of our comparisons. And for that, we used a false discovery rate correction that is a bit more lenient than a standard like Bonferroni, where you just multiply the p-values by the number of tests you're running, but it's still statistically valid. That can let us find some interesting trends. And then there are some where I do an ANOVA, and then I do the standard post hoc Tukey test to compare different pairwise treatments. All right, so actually getting into the data, the first thing I'm going to be showing here is the internal ethylene. So as I mentioned, we expect AVG to decrease ethylene production. You see here, just looking at air storage, these gray points, we definitely see that it does, that for when ethylene is not added, we see it starts higher and rises very quickly, but when AVG is added, we see that it stays much lower than the control. And then when we look at the controlled atmosphere treatments, here we see that a controlled atmosphere does keep ethylene levels lower than in air storage, which we expect, and that even within these treatments, these circles here have higher ethylene than the triangles that represents the AVG applied. So basically, this is telling us exactly what we expect to see. It confirms that, like, yes, our controlled atmosphere and our AVG did work how we thought it would. AVG reduces ethylene, controlled atmosphere reduces ethylene. All right, and then going into the actual microbial data, I know a lot of people don't have a ton of experience reading these ordination plots and working with these huge multivariate data sets. So I think it's a good idea to walk people through kind of what these mean and how to read them. So a couple of key points to understand are each point here represents a microbiome of one sample. So the 10 apples that we put in a bag, get the bacteria and fungi off of, each one of those samples is 1 point on here. And a lot of people, when I show these graphs, ask, okay, well, what do the axes mean? If something's higher on the x-axis or the y-axis, what does that mean? And the answer is it doesn't intrinsically it's not intrinsically measuring anything except just the distance between the points matters. All right, so that when we have this huge multivariate data set, you can't always get like a super intuitive, like, oh, this is a better microbiome versus a worse microbiome. All you can do is say, with these huge number of variables we have going around, we can collapse them into 2 dimensions, and the points that kind of cluster together and that are more similar, are closer to each other in this multidimensional space, have microbiomes that are more similar, right? And so the way to read these is to look for certain clusters of microbiomes that cluster by or different treatments. And in here, we don't really see anything. It looks like the points, colors, and shapes are just kind of scattered randomly, and the stats line up with that. Then when we look at these fungi here, we don't really observe that our different storage types, storage times, or AVG treatment impacts the microbiome. And that when we look at bacteria, on the other hand, we do see a bit more clustering. And to me, the way I would describe this, trying to make it as simple as possible, is the points of different colors are kind of clustered together. They're more closer to each other in space than they are to the other points. And we see that the stats tell us that storage type and time, which we're using as just one variable here since our sampling was a bit complicated, and the AVG treatment are both significant predictors of microbiome variation. And visually, the way I point this out, not that the circle is based on the stats, is that these points here, these dark red and blue points that represent the 9 months time point for both our controlled atmosphere, our 0.5% and our 2% oxygen treatments, are pretty different, pretty far apart in this multivariate space from all of these other points. So my main takeaway from this is that after 9 months we get of low oxygen storage, we get microbiomes that look pretty different from most of the other time points. All right, and then going into results next, for fungi, we didn't find any difference in the composition of microbes. We also don't really find any difference in the diversity, which is how many fungi there are and how equally they're spread. Right, so all these points are, we don't really see any significant differences. We also don't see a difference with AVG. But with bacteria, we do. We see that with this long-term low oxygen storage, you see the diversity goes down a bit. AVG doesn't affect it significantly, but storage time/type does. If I had to like try to interpret this, my best guess is that there just aren't that many different types of bacteria that can survive 9 months under this is 0.5% oxygen storage. So because of that, we see that the diversity is lower because there aren't as many different types of bacteria that can survive there. All right, and so now I'm going to go into talking about what I did with the predicted gene pathway data. And I was told to warn everyone that the stuff we've dealt with before is not necessarily like super basic, but this can get somewhat technical, and it's important to understand kind of what this data is and what the data isn't. So what we're doing is we're taking these amplicons. What we have are these tiny DNA regions that are sequenced. We're matching them to reference genomes, and we're saying, okay, what gene pathways do these reference genomes have? We'll add these to our big table of different counts for gene pathways, and then see how those vary with our treatments. Right, and so in this case, with the example, if we have 5 different DNA strands that match up with this reference genome, we would say take all of these, take all the gene pathways on here and add 5 counts in our big table, and here the same with these 2 strands that match up to this second reference genome. Right, and then another reminder, looking back at our sampling schematic, I was really interested in comparing between air and our controlled atmosphere. And the question is, well, what time points do you compare? You can compare them all when they're at 5 months, but in that case, those air apples are really late in storage when the controlled atmosphere apples are still earlier in storage. So you get differences in physiology there. You can also compare them when they're both early in storage, but then maybe the fact that the bacteria have had more time to like build up for 5 months in the controlled atmosphere can impact the results. And then same when they're both late in storage. So here I have, basically when I do these comparisons, I'm doing 3 different comparisons. 3 months air versus 5 months controlled atmosphere, 5 months air versus 5 months controlled atmosphere, and 9 months controlled atmosphere versus 5 months air. All right, and basically what we see is that this comparison between controlled atmosphere 9 months, kind of very late in storage, when we have low oxygen compared to as late as we can store air apples, we see the biggest differences. So on the left here, we're looking at the different genera, and we see that about 30% of them change in abundance with this comparison, looking at 9 months controlled atmosphere versus 5 months air. And then over here, when we look at the gene pathways, we find that it's also about 30% that increase or decrease significantly with this comparison. And so the question then arises, all right, so we have all these gene pathways that are different between this comparison when we look at 9 months very late in storage with atmosphere versus 5 months air storage, also late in storage time. So what are these gene pathways and what can they tell us? And the trends that I was able to find, big ones. The first one that stuck out to me was that a lot of the gene pathways that decrease in abundance in the controlled atmosphere, in the low oxygen, are responsible for aromatic degradation compared to degradation pathways and all pathways in general. And I'm guessing some of the apple people are, when you hear aromatics, you think about like esters and apple aroma compounds. The tool that I used for this defines aromatics using the organic chemistry definition, so compounds with benzene rings. So the translation there isn't quite one-to-one, so the interpretation is a little more tricky. And then over here, one of the other striking things I noticed is that the pathways that use oxygen as a reactant, when we have long-term low oxygen storage, those pathways tend to be decreased in abundance, which is fairly intuitive that if you have very low oxygen for a long amount of time, the bacteria that are selected for probably have fewer pathways for using oxygen. And at this point, I took a step back and said to myself, okay, well, like I want to make sure that because this metagenomic data is so complicated, I want to make sure that I'm not making a whole story out of nothing. So I compared oxygen, which again functions as a final electron acceptor during cell respiration, and in general has many other roles. So as an electron acceptor with other common electron acceptors, and found that those ones didn't share the same trend. Though a caveat is that like I'm not a trained like cell biochemist. If there are other interesting compounds that oxygen should be compared to here, I'd be happy to hear them. But this basically suggests that oxygen, but not compounds that have similar roles, are decreased in long-term low oxygen storage, the percent of pathways using oxygen as a reactant. That's confirmed as we go through, and instead of looking at how many pathways significantly increase or decrease, looking at, okay, let's take all the pathways with different compounds as a reactant and add them all up, that we see that these couple of this dark gray and black point here representing air storage, about 10.5% of the pathways that they have use oxygen. And for this long-term 0.5% oxygen storage point, it's about 9.5% of pathways. And then that difference is statistically significant. And that with the other common electron acceptors that we observe, we don't really see the same trend. That here, these long-term low oxygen time points don't decrease in NAD+, and they also don't decrease in NADP+. So it does seem to hint that there's the strong potential for the low oxygen treatment to be impacting the microbiome, okay, by selecting against pathways that require oxygen, which is very exciting because a lot of the microbiome studies, because we don't get species level identification, aren't really able to make those claims. But I also do want to like take a step back and say that because we're looking at, again, what's the equivalent of like an entire forest, just microscopic, it's important not to like overinterpret from this data. And there are some aspects of it that make it so that I don't feel comfortable definitively saying this is the case. The biggest one is that at 5 months time point, when we look at the different treatments, we don't really see any difference when they're all at 5 months. At 9 months we do, but at 5 months we don't. So is that low oxygen is selecting on pathways that require oxygen, but it just takes a long time for us to see real results? Or is it the fact that we're able to store these apples for 9 months, that there's something else about that that decreases the abundance of these pathways that happen to require oxygen? It's kind of unclear right about now, but I also think it's exciting that we're able to tie this potential for this functional explanation of oxygen and pathways requiring it to these microbiome composition changes, which a lot of similar studies haven't quite been able to do. Right, and so that was kind of technical. Let's zoom back a bit and summarize what we found here. So the big points are we found that our storage time and type, low oxygen storage time, as well as this ethylene inhibitor AVG treatment impact the bacterial microbiome, but we don't really find any evidence that they impact the fungal microbiome. And those impacts on the microbiome, we also find that pathways using oxygen and pathways for degrading aromatic compounds are decreased in abundance late in storage for a controlled atmosphere when compared air storage. And there's the potential for that to be related to the lack of oxygen selecting on the community, but it's not super conclusive at this point. All right. And then I'd like to say thanks to my advisors in my committee, all the people that I've collaborated with, not necessarily on this project, but in a lot of the work that will be going as my PhD. And then present and former lab members that helped me in the way, and then also my funding sources. Thanks for listening.
All right, let's open up for questions.
Greg?
Connor,
Mhm.
So does the apple itself matter in the microbiome that you measured? In other words, if you just had like a wood block and you covered it with wax and then inoculated it with bacteria, do you think you would come out with similar results, or do you think that there's some interaction between the bacterial community and the apple that is not, you know, clear from what you measured here?
Yeah, that's a great question. And I will say, like, however I answer that is,
We have to repeat the question.
oh yes, yes.
Yeah.
So the question is, to what extent does the fact that these microbes are on an apple surface actually matter? If they were on just any sort of other surface, would we see the same results? And the answer is, there are a lot of studies that look on microbial communities of surfaces, like not usually like wooden blocks necessarily, but like on the walls here. And we do see some differences in microbiomes between, say, like what is on the wall or what is on the chair versus like what we would find on an apple. In terms of specific aspects of why being on an apple would drastically affect the microbiome compared to another surface, that's one of the things that, to my understanding, the field is still like working on establishing like what the actual kind of key relationships are. That more than likely the plant microbe interactions do have an effect on the microbiome, but characterizing exactly what those are, because there are so many different microbes and we don't know what all of them are doing, is a bit tricky.
So you mentioned the potential of ethylene to be something that's changing the microbiome.
Mhm.
Do you plan on looking at that interaction? And if so, how would you know that it's the decreasing of ethylene itself rather than the addition of this compound?
So yeah, the question. So the question was, like, well, what would I do to distinguish between the fact that this compound is being applied versus the actual reduction in ethylene itself? And there are like a few ways you can potentially do that. Like there is a compound called ethylene that like where you can basically artificially add ethylene. The way a lot of people in the apple community see it is that the variation due to when you apply this chemical, the effect it has is like so strong in terms of ethylene that it is like a pretty decent stand-in, that it's has a huge effect on the ethylene levels, and that it is somewhat regularly used as like a stand-in for, we apply this compound, ethylene decreases, we see these effects, most likely due to ethylene. One thing I will say is that when I did the multivariate analysis for whether or not like ethylene levels of ethylene itself affect the microbiome, it came back negative, which is most likely due to the fact that like most of the variation we see in ethylene is due to this compound. So kind of small bits of variation within that we saw didn't really affect the microbiome. So that's kind of my answer, is that like the compound is used as a stand-in for something that just decreases ethylene. Those big differences affect the microbiome. We don't see the same thing for the small natural variation when we don't use the compound.
Time for one more or no? Time for one more. Yes.
We get to 20th or 15th.
Oh,
after.
okay. Time for many more. Lucky Connor.
I only have one sort of.
Mhm.
This might be similar to Greg's question, but do you or do folks know where the majority of these species are coming from in terms of you're looking at them in the storage setting? And then I guess with that, like, do you have an idea as to why the stem makes sense to me, but seeing that seeds also have like significantly more bacterial species than other parts? Do you know why that would be, or have an idea?
So yeah, the question is, So can you repeat the first part of that?
Yeah, just like, are most of these species originating in
Oh,
the field,
mhm.
Harvest equipment?
Yeah,
Where
the
they're
question is, where do these species come from? And the second part is, we saw that the seeds have a lot of different, have a very high counts of bacteria compared to some of the other areas of the apple. And why do I think that is? So for the first part, where do these microbes come from? It's not super clear, and that's one of the things that I am hoping to work with on my PhD, is looking into where these microbes actually come from. There's been some work in the field trying to distinguish, okay, like how does dispersal actually happen? Some people have suggested it's due to soil, the fact that the wind picks up microbes from the soil and deposits them onto these above ground areas. That's the key factor. Other people have said, well, maybe it's like rain or insects or so and so. When we look at post-harvest, when we do, when there are some basically statistical things we can do to assess, okay, are the changes we see due to the microbes that are already there just increasing and decreasing in abundance, or are there new microbes being added on as well? And we do see that there are new microbes being added on as well. In ecological terms, there is turnover. There's not just nestedness is the term we would use. The question of how exactly does that happen, we're not totally sure.
Want to ask Geneva, they have questions?
Yeah, Zoom, are there any questions? Maria?
It seems like from your data that there weren't a lot of differences between for the fungal communities,
Yeah.
but it also sounded like in the beginning you guys were talking about spraying fungicides as post-harvest control of diseases. Like, do you have and also like products people that are commercially available for disease control? And you're talking about how that's not super effective right now, but do you have any thoughts about like why we use fungicides for disease control for fungal diseases, but yet you're not actually seeing differences in your fungal communities based on your...
All right, so the question is, we don't see that the fungi in my experiment, the microbiomes change with a lot of treatments. And the question is, okay, in that context, why is there so much emphasis on these fungicides if we see that these fungal communities are kind of hard to change? And so, kind of two-part answer to that question. First is, and I think this highlights that some of the complexity of these microbial communities. This experiment, we didn't really see any changes in the fungal communities. Some of the other experiments we definitely have, and when we test different factors. And then the other point is, the best fungicides, in my mind, are ones that don't necessarily have these huge impacts on the entire microbial community. That if we're targeting a specific pathogen, you spray the fungicide that targets that one pathogen, that leaves the rest of the microbes untouched. There are some people who have postulated that like that's a good thing for like fruit health, that if you're not changing the microbial community as much, then that can be better. But another big concern is that if you're using these like multi-site fungicides that are more likely to kind of disrupt all the fungi, those also have a bigger risk of just being harmful to humans. The more specific you are in targeting the one fungi you need to target, the lower risk there is of it kind of going awry and targeting anything else that you don't want it to target. Yeah.
My question is also about your fungal data. You mentioned that it was hard to get really great resolution. And this is kind of asking you to speculate, but I wonder if you have a sense of whether, you know, the fact that you weren't able to see shifts in the fungal microbiome had to do with the fact that you didn't have a very fine resolution. You know, maybe there were species shift, but because you were only looking at genera or something, you didn't see those shifts.
Yeah, the question is, I mentioned that we don't always have species level resolution. So like, could the fact that I didn't see any difference in the fungi be due to the fact that there were shifts, but just on a level that we couldn't detect? And the answer is that potential is absolutely there. I mean, if it were happening, I would have no way of knowing just because I can't detect it. My best guess is that those shifts are not going to be the most impactful. That a lot of times, and again, it's like hard to compare because there aren't a ton of studies that really go in with these kind of deep, high resolution sequencing. So it's kind of hard to compare. But we see a lot of studies both on apple surfaces and on the surfaces of other fruits like grapes, and then also in soils and in humans, where we are able to detect a lot of really substantial differences in the fungal communities based on whatever treatments we apply. So to me, that seems to imply that like the lack of resolution may be an impact, but there's still the potential to see lots of big differences here. And I wouldn't say that the resolution issue is the biggest reason why I think we didn't see differences, if I had to guess.
Anybody else?
I got one more for you, Connor.
Ah,
So you introduced the topic about the human microbiome and our gut microbiota. So do you eat, right? So do you think that there are human health differences based on the apple microbiome in terms of the different types of storages, in terms of what we can expect for human health?
So the question is, because I talked about the human microbiome, do I think that the differences in the microbiomes I find on the apples could also have a human health impact when looking at the human microbiome? And the answer is, there have been, I've heard people feel very strongly both ways. That I've heard some people in these fruit microbiome studies being like, yes, like it's really important to like know what microbes are on there because like these could be super impactful for human health. And then we see that human and that gut microbiome does depend on diet is a pretty well established thing. But then the other side of the coin is people saying, well, like most of these microbes are probably just getting destroyed by stomach acid anyways. So we do see some differences based on diet, but that's more likely to be due to like the nutritional composition of what goes into the gut and the actual microbes that came in. Of course, that itself is complicated too, because it's not like not all of our microbes are being destroyed by stomach acid, because stuff like food poisoning still happens from microbes based on stuff we consume. So my answer is basically like, I think it's a possibility, and that's a very contentious question right now.
All right, well, please join me in thanking Connor for.
All right, thank you very much.
This has been a production of Cornell University, on the web at cornell.edu.